So we are talking about this diplotin and diakinesis. Now see here, this is the stage where we can see that the chromosomes which are present at the parent cell at the beginning, okay, we will see the same number of chromosomes in prophase 1 and diakinesis, same number, 4 chromosomes. Okay, here 4 are there, here also 4 are there. 23 pairs are there, here also 23 pairs are there. But what is the change? The genetic combination which is present in this chromosome is totally different. Okay, is totally different because please mute yourself. So totally a new genetic combination is seen. So once again, we'll just have a look at this stage. Okay, meiosis 1 and meiosis 2. There are two different stages in meiosis. The number of chromosomes are present in the prophase 1. The genetic combination has taken place. Okay, so we already know that what are the stages of prophase 1: leptotene, zygotene, pachytene, diplotin, and diakinesis. Then the cell enters into meiosis 1 stage, next stage. See here, you can see a combination prophase 1. Prophase 1 here, what we can see is that the nuclear membrane, again, same like mitosis, we'll see that the prophase 1 nuclear membrane is completely removed off. And can you see the metaphase 1? The new genetic combination is seen. Okay, we can see that a new genetic combination of each chromosome. Okay, so then what is happening later on in anaphase 1? Again, spindle fibers are there, but here what is happening? There is a difference in the splitting of the chromosomes. Can you see that here? The 23 pairs of chromosomes are there. The spindle fiber is attached to the entire kinetochore. Okay, total kinetochore, not at the center. Half of the centromere is not attached here. The total centromere is adhering to the spindle. Okay, this is a speciality which is present only in the germ cells. So because of that, what is happening when there is splitting of the anaphase, splitting of the cell at anaphase 1? What is happening? The chromosomes are reduced to half. So what is happening? Each daughter cell at the end of meiosis, guys, can you see the presentation?
No,
Yes,
ma'am.
ma'am.
No, no, ma'am. Yes.
Yes, ma'am.
Yes,
Ma'am.
yes,
Yes,
ma'am.
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 Yes, ma'am.
Ma'am, but we can't hear you.
Wait a second, please. Can you all see the presentation?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, so can you all listen?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, fine. So we are discussing about the meiotic stages. So prophase 1, metaphase 1 and anaphase 1. What is actually happening here? Properly, the cell divides, but remember that we can see that the number of chromosomes is reducing. So this type of division is called as reduction division. Okay, so it's a very important word. Please keep in mind what is this type of division called as reduction division. Why it is reduction division? The genetic material which is present inside the chromosomes is split. Combination of maternal and paternal genes are present, and the number of chromosomes are splitting into half. So each daughter cell is getting only half of what maternal cell is having. So here you have 23 pairs of chromosomes in the maternal cell, but in the daughter cell you get only 23. Okay, so 23 chromosomes in one cell and the other cell contains 23 chromosomes. So instead of 46 we get only 23 chromosomes in each daughter cell. Okay, so this type of division is called as reduction division. Very important statement, please keep in mind. So now once this is done, it goes to meiosis 2. Meiosis 1 is a very important phase. Okay, so you have to explain all the stages. And when it goes to prophase 2, from here onwards, meiosis 2 is just like mm, mm, uh, mitosis. There is no difference. Same like mitosis, the nuclear membrane disrupts. Now, the whatever is present, the number of chromosomes, only 23 are present, not 23 pairs. Only 23 are present. So this 23 will have DNA replication. Then you will see that spindle fiber formation in metaphase at the equatorial plate. Anaphase, the spindle fibers split this the chromosome into half so that each daughter cell gets same number of chromosomes where prophase 2 cell contains. Okay, so here at the end what we are getting, total 4 cells. Out of meiosis we get 4 cells. One maternal cell will give rise to 4 daughter cells in meiosis at the end. Okay, but here what are the main changes? In first cell you have 46 chromosomes. At the result of meiosis we get 4 daughter cells, each containing only 23 chromosomes in each cell. Okay, so this is very important. Also remember that the chromosomal genetic combination is changed. Okay, genetic variety is changed. So you will see that the genetic material of the daughter cell is completely different than what the maternal cell is having. Okay, so it's a new type of cell. So this is the speciality in the chromosomes of meiosis. So this is what is actually happening. You can see at the centromere how the chromosome structure is split into two. Okay, so here you can see the tetrad formation, chiasma. This pachytene, during pachytene we have understood that, right? Chiasma formation, these are the patches. So this patch is called as chiasma. Okay, so non-sister chromatids, one is from the maternal side, one is from the paternal side, they are joining at the patches, and because the combination is going on, see here a new type of chromosome is seen with a new genetic combination. Okay, sorry, can you see the presentation?
Yes, ma'am.
Yes, ma'am.
Yeah, Then you go for metaphase 1 and anaphase 1, telophase 1, and then go to the prophase 2. So here you can see the cell membranes very lightly. This is an animation. So metaphase 2, same thing, anaphase 2 and telophase 2. So as a result, we get only half the number of chromosomes. Now, where does this meiosis happen? Meiosis happens in the germ cells. What are the germ cells? The oocyte and the spermatocytes. Okay. As a result, what is happening? One germ cell gives rise to four daughter cells. Four daughter cells as a result of meiosis. Now, there is something which is little bit more addition than what is meiosis you have learned till 12th. What is happening here? There is unequal division of cytoplasm. You see in the oocytes. This is the speciality in female. What happens is all the cytoplasm which is present, the number of chromosomes are splitting into 23 and 23. Okay, but the cytoplasm is completely entering into only one cell. At the end of meiosis 1, we get two daughter cells, okay, with 23 chromosomes. But the cytoplasm, look at the cytoplasm, all cytoplasm is taken up by only one cell. The other cell gets only a thin rim of cytoplasm. Okay, so this is called as polar body. Polar body. Now, this is not functional. This cannot be fertilized. What can be fertilized? Only secondary oocyte can be fertilized. Okay. Now, this again undergoes second meiotic division, gives rise to mature oocyte and another polar body. This polar body also will divide into two. Division happens, but remember that these polar bodies are non-functional, cannot be fertilized. Even sperm goes inside. It can also form male and female pronucleus, but still it cannot give rise to a zygote because there is no cytoplasm. So not just genetic material, we require entire cytoplasm also for the formation of a new human. So what is required? The cytoplasm and the nucleus. So this is called as unequal division, unequal division of cytoplasm. So as a result, what is happening in the females? We get only one mature oocyte from one primary oocyte. Okay, so that's why only one oocyte goes for ovulation. So as a result, only one cell is getting fertilized. Okay, but in lower animals, you see dogs, cats. Okay, so what is happening? These are still remaining. Okay, so number of oocytes are more. So as a result of one division, it gives rise to four, sometimes six. In dogs and all, you see more. So these all get fertilized at the same time. Okay, as a result, they'll give birth to multiple babies at the same time. But humans, we are programmed to get only one mature oocyte out of one primary oocyte. Got it? So in case of males, that's not happening. More number of sperms are required. So what is happening? One primary spermatocyte give rise to four spermatids. Okay, so here equal division of cytoplasm is happening. So as a result of first meiotic division, you get secondary spermatocytes. As a result of second meiotic division, you get four spermatids. Okay, so this meiosis is happening only in the germ cells. Okay, oocyte and the spermatocyte. We'll discuss this more in detail in the next embryology lecture. Okay, right now you just concentrate on only what we are talking about. Okay, what you need to remember is what is meiosis, what are the stages, where does it occur, what is the difference in male and females. Okay, that's it. Till here clear, everyone?
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, now why it is important for us to learn? Okay, let's go to the abnormalities. Sometimes what happens because of the genetic recombination. So see here what is happening. We get 46 chromosomes and a genetic recombination is happening. Normal meiotic division happens like this. Okay, first meiotic division and second meiotic division. So we get 23 chromosomes in single cell, right? This is a normal meiotic division. But sometimes there can be abnormal meiotic divisions, right? So abnormal meiotic divisions are called as disjunctions, okay? Disjunctions. Now see here, non-disjunction in the first meiotic division, because what is happening while splitting, the spindle fibers have to take the chromosomes to each side. So what happens because of abnormal division, all the chromosomes are entering into one cell and the other cell does not get any chromosomes. As a result, second meiotic division contains now more the chromosomes. Okay, right? Not all. Sorry, not all means what do you mean by this? See, 23 pairs are here, right? So 23 should be there. Instead, what is happening because of disjunction? Okay, out of 23 there might be one extra chromosome going into another cell and one is left out. Okay, so as a result what is happening, these cells coming from second meiotic division might have an extra number of chromosomes. So they might have 24 chromosomes. Okay, while other set of cells can have only 22 number of chromosomes. Okay, this can happen at first meiotic division or else at second meiotic division also can happen. So as a result,
Ma'am, can you repeat it once again?
yeah, see, let's say these are 23 pairs of chromosomes, okay, maternal cell. Now this is undergoing first meiotic division, gives rise to 23 here, 23 here. Second meiotic division, 4 daughter cells with only 23 chromosomes in each. Sometimes
Yes, ma'am.
we'll see non-disjunctions. Non-disjunctions means abnormal splitting of chromosomes. So what happens? This can happen in the first meiotic division or second meiotic division. In first meiotic division, if it is happening, what will happen? One cell is getting one extra number of chromosome and the other cell might lose a extra chromosome. Instead of 23 and 23, we get 24 and 22. Okay, so in second meiotic division,
Okay.
it might happen same like this. So what is happening? One cell will get 24 and another cell will get 22. Okay, so that means, see why it is important. Let's say this is maternal cell, okay, and paternal cell, that is sperm. This is one egg and sperm. They are fusing, and we are going to get a zygote after fertilization. So total number of chromosomes in zygote, tell me if it is 24 here. What will be the total number? 24 plus 23, right? Instead of 46, we'll have
Yes, ma'am. 47.
47 chromosomes. Okay.
47.
If this cell is fertilizing with a sperm, then we will get how many number of chromosomes? We get only 45 number of.
45.
Yes. So these are called as disjunctions. Now see here, 46 chromosomes, normal cell division should give 46 and 46 after fertilization. Now abnormal cell divisions, they are fertilized. So what will happen? Either we might get one less or one more. So either we might have 45 or 47 chromosomes. So then what? If this cell is growing, okay, because of that, what happens? Multiple cells are coming now out of each cell. So see here, can be extra number of chromosomes. This is called as mosaic trisomy. Trisomy 21. Trisomy 21 means what? 21st number of chromosome, we have 3. Okay? Instead of 2,
Yes, ma'am.
3. Right? See here, you might think just one chromosome, how does it matter? How does it matter? One extra chromosome, how does it matter? But see here, Because of that one extra chromosome. We are taking one example, only trisomy 21. 21st pair of chromosome, instead of pair, two chromosomes, one maternal, one paternal, we have one extra, okay, on 21st. So see the child, this child, trisomy 21, we get Down syndrome. Down syndrome, what are the different changes? See, flat, broad face, palpebral fissures or oblique, protruding tongue. Okay, usually intellectual disability, many cardiac defects, broad hands, and single transverse crease. So all this like this transverse crease, only single transverse crease. Okay, webbed fingers. We'll see so many different changes because of one chromosomal abnormality. Okay, now see, this is trisomy 18. Means trisomy, when I'm saying trisomy 18, that means what? 18th chromosome. Instead of 2, we have 3. See what kind of changes you will see. Okay, low set ears, ears are small. Okay, small mouth. Okay, deficient mandible, this is called as micrognathia. Okay, flexion of the hands, absent or hypoplasia of radius and ulna. See, no forearms. This is trisomy 13. 13th chromosome, we have 3 in number. Bilateral cleft lip. See here, I told you, cleft lip and cleft palates. Sloping forehead. Anophthalmia, no eyes, no palpebral fissures. Usually the child is difficult to survive. Okay, now Turner syndrome. All these are various chromosomal abnormalities. See, one small chromosomal difference, you will see lot of changes. Turner syndrome, right? You'll see lot of changes. Webbed finger, short neck, malformed ear. See here, you can see webbed neck. Okay, see the foot. Angelman syndromes. Okay, maternal chromosome, microdeletion. Microdeletion means one less chromosome. Okay, that is we have only 45 chromosomes. So we'll see Prader-Willi syndrome, very painful syndrome it is. Okay, paternal chromosome is, see, it can happen on both maternal or paternal. If it is usually microdeletion maternal chromosome, okay, if the paternal chromosome is gone, it is called as Prader-Willi syndrome. Okay, maternal chromosome microdeletion. Maternal chromosome deletion is not so much difficult, but paternal, it is very difficult. Okay, see, this is how plating we do it. Okay, when chromosomes are, this is a karyotype. See the Down syndrome. See, these are the numbers which we give to each pair. Okay, now see 21st chromosome Down syndrome, there are 3. You can see that. So this is the trisomy 21, right? So how we can find out? We use, usually we do just by karyotyping, or we can do fish technique. Okay, immunohistographical technique. So here when we use this technique, what happens? If any cell which has got extra number of chromosomes, they are highlighted in red color. See here, here we can see 3333. Okay, chromosome 21. Okay, chromosome 21, we have 3 in number in each cell, right? So like this, we can use various techniques. So that's all for this. Now these are the questions which can come. Meiosis, we have to write under following headings: definition, location, process, and clinical application. And questions are there, multiple choice questions, you can attempt them. Okay, I had given all the questions, multiple choice questions to you, so you can attempt. So out of this chapter, you have any questions to ask, you can ask me. Any questions?
Ma'am,
Yes.
Ma'am, the heart cell is present in the G0 phase, right?
Hmm. Yes.
But but you said that it is present in the G1 phase and it
No, see,
performs a normal growth.
listen. G0 and G1. Okay. Whenever it is required to grow, then it enters into G1 phase. Some cells enter into G0 phase. They doesn't grow, they are remain just like G0 phase only. But from G0, it can go to Z1 always. Remember that. Okay.
Okay.
Yeah. Any more questions?
Ma'am, can a stem cell produce a stem cell like
Yes.
yeah.
Yes, definitely. Because stem cell property is that it gives rise to one parent cell, which can again divide and give rise to, it's called as like a basic population. So the basic cells will remain, but the other cell, which, if, for example, like let's take spermatogonia. Okay, here. Spermatogonia are there. Before coming to primary spermatocytes, spermatogonia are there. So many spermatogonia. So they keep on dividing and gives rise to type B cells, type A, type B cells. Type A cells always remain as germ cells or stem cell. It keeps on dividing into type A and type B, type A and type B. All type A remains as type A's, and type B's convert into spermatocyte, primary spermatocyte. Okay, they are more differentiated. So stem cell property is that they keep certain kind of basic population always present, and they gives rise to cells which can differentiate on requirement. Okay?
So ma'am, a totipotent cell will give rise to a totipotent cell?
Yes, of
Okay,
course.
okay.
But to a limited quantity only, na? Only till limited quantity, only till morula stage. After morula, you will not get any totipotent cells. Okay?
Yes.
Any more questions?
Ma'am,
Yes.
in test there was one MCQ
Hmm.
in which it was written that the hip is proximal to the knee.
Hmm. Yes.
So that was the right answer, but the knee is proximal to hip wasn't right. So like, what's the difference between both the sentence?
Proximal means what?
Nearby
Hmm.
means near to body.
Not near to the center. Remember that. Okay?
Hmm.
Okay. So hip is more proximal. Okay. Proximal means upwards, closer to the center. Distal means away from the center. Okay. Knee is more distal to the hip, right? Hip is proximal to the knee. Proximal means above or upwards. Okay.
Yes, yes. Thank you, ma'am.
Hmm.
Ma'am, what is chiasmata?
Sorry.
Chiasmata.
Chiasmata is the structure where we are. Chiasma means crossing. Okay, crossing means where exactly we'll see that the chromosomes are crossing. Okay, so that is a point where the two non-sister chromatids are crossing with each other. So that is a area where we say that the patches are formed. Okay, that is where the crossing over takes place.
Okay. And ma'am, what is external flexion?
External flexion, I told you, right? It is something like just like extension, synonymous to extension. Okay.
Okay.
Any more questions? So at the end, I want all of you to just drop in your feedback. Are all of you understanding? Are you enjoying our lectures? Please drop the feedback today. I'll just send the link in Google Classroom for the feedback. Please give your feedback in Google Classroom only. Okay? I'll send the link to you separately. Only in that, please try to send your feedback. Okay? Any more changes
Yes,
you
ma'am.
would like to do, or any more more details you want, or some of you may not understand what is happening, so we can communicate at the beginning stages, or you might have missed some portions, so please let us know so that we can correct it at the beginning itself. Okay? So are you enjoying? I hope you're all enjoying ANAT lectures.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Definitely.
Yes, ma'am.
Good. So I'll take your leave then. Okay. I have already given the PDF. Now I am trying to give this links to all the videos, whatever we had taken the classes. Most of the videos are there. Maybe one or two might be missing, but rest of the videos, I'm trying to make one folder and I'll uh, uh, submit to all of you the link to that folder so that you can get access to all the videos, whatever classes we have conducted till now. Okay. Someone asked, where are we supposed to draw cell cycle? Guys, you can make your own notes. Okay, whenever you are writing, whenever the lecture is going on, make a note to yourself so that in exam also, if a question is coming, you know what diagrams to draw. Okay, cell cycle also draw it on a normal notebook. I'm not saying it's not an assignment, you can draw and learn. Okay, but remember one thing, we have an assignment for the next week, that is charts of mitosis and meiosis. Make beautiful charts, take a picture and submit in the Google Classroom again. Okay, so today I'll be sending the link for that as well. And please submit your assignment
Ma'am.
cell model, just a second, cell model by max to max by 1st of January. Okay, chart can be any size, dear, no problem at all. Normal, wall mountable charts are very nice. We are not, it's not compulsory, you can draw it on a normal drawing paper also, it's okay. You just need to submit, that's it. Okay. If some of you had made beautiful charts, we will select some charts and we'll keep it in our histo lab, so that later on whenever you want to walk in and see yourself, all your charts, you can come and see, no problem. Okay. Yes, someone is asking a question. No more questions?
Ma'am,
Yes. Yes, Sakshi.
ma'am, we'll be getting the PDF through GS, sir, right?
Right
Today's
now,
PDF.
as of now, I had already given this PDF, right, yesterday itself.
Yeah, ma'am. The previous PDF we got it from sir.
Yes, yes. This also, the entire presentation is a continuous one, and I had given that PDF to you,
Okay.
right? Yesterday itself.
Okay.
Yeah. Any more
Okay. Thank you,
questions?
ma'am.
Okay, then. So please keep yourself updated. Check the Google Classroom for the links. Okay, two links I'll be sending. One is for the submission of next week's assignment and a feedback link. So please give your feedback in that. Okay, so that's all. And I'll take your leave. Have a good day.
Thank you, ma'am.
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 Thank you, ma'am.